Hello, and I want to thank you very much for the opportunity to present our work today. I work with Jim Palace, and we're part of Hub 2 with PI Stella Chow. And our overarching goal is to develop reagent red cells using genetically modified iPS cells, which will allow us to express specific blood antigens to better type blood, which will be especially helpful for our sickle cell patients. And we hope also will provide some insights to help with a long-term goal to make transfusible cultured RBCs. In order to do this, we have to accomplish three things. We have to take iPS cells and differentiate them into erythroid progenitors. And then to take those erythroid progenitors and use them to expand a pool of erythroid precursors, which we can then use to mature to make functional red cells. And I'm going to talk about each of these three steps today. Now, first, there's a complication with making erythroid progenitors out of iPS cells that, as seen in many other systems, iPS cells recapitulate fetal development. And during human fetal development, there are multiple erythroid lineages that are formed. First, there's a primitive erythroid cells that are rise in the yolk sac and provide the first circulating RBCs. But then there's a second independent wave of erythroid progenitors that are part of an erythroid myeloid progenitors, or EMP wave. And they move to the fetal liver, which is the initial site of hematopoiesis in the embryo, and they make definitive erythroid cells. And then a few weeks later, they're followed by HSCs who have formed in vascular beds and also move into the fetal liver and also make definitive erythroid cells. Now, we know we can use the expression of beta globin genes to help distinguish these different types of erythroid cells. Primitive cells express mainly epsilon, beta globin, or HBE, and definitive cells from either EMP or HSC express predominantly gamma globin, HBG. And then in the adult, the HSC switches to using beta, or HBB. Importantly, our collaborators have shown it's not just globin genes that are differentially expressed, but primitive erythroid cells express a subset of the cytoskeleton proteins that we find on our definitive erythroid cells. And this includes missing some of the key antigens that we want to use for reagent red cells. So we'd like to start with definitive erythroid progenitors for this project. We selected an iPS differentiation system that was based on one published by Olivier in 2016, because we know they ended up with cultured RBCs that had predominantly fetal proteins for globins. So we reasoned there had to be a definitive progenitor earlier, although there could also still be primitive. And we found that that was actually true when we turned to a process of looking at individual cells' RNA through RNA flow. We're able to show up here on the top left that primary fetal liver, you can see cells that predominantly express gamma globin. In bone marrow, you can see beta. In iPS, we saw two types of cells: primitive cells that expressed HBE, a high amount of epsilon, and fetal that expressed a high amount of gamma. And they occurred in different proportions over time. So what we did is we cultured progenitors from different times during that differentiation and found that there was a peak of definitive progenitors at day 12. We further looked to see if we could enrich in definitive versus primitive erythroid potential in subpopulations at day 12. For this, we looked at CD43 positive cells, and we found two populations. One is double negative for CD41 and CD235A. That population actually makes predominantly myeloid cells, but the erythroid cells it makes are definitive. And then there was another population that had that was 235 positive, and it expressed 34 as well as 45. And it's a rare population, but it is very much enriched in definitive erythroid progenitors. So what we did is to take day 12 and those subpopulations, and that's what we used to move on to the next step. Now, in order to expand erythroid precursors, we turned to this window of opportunity during erythropoiesis, which is hallmarked as this extremely expansive system. But between late progenitor and early precursor stage, there's a sensitivity to erythropoietin and glucocorticoids that allows for an expansion of self-renewal of those cells during anemia. And if you use those signals of erythropoietin, stem cell factor, and glucocorticoid, dexamethasone, many groups have shown a short-term expansion of these early precursors in vitro. We had done work in the mouse years ago to show that we could actually get extensive expansion with those three signals out of the mouse, and that this relied on an increased expression of BMI1. BMI1 is a member of the polycomb repressive complex, and it's been found in a number of different stem cells as well as malignancies to be involved in self-renewal. And we found that even in the high, if we kept high expression at BMI1 though, if we take out dexamethasone, these expanding cells would still be able to complete maturation, make RBCs, both in vitro and in vivo in the mouse. So to apply this to the human system, Jamie Olson in our lab used a lentivirus to overexpress BMI1 in peripheral blood mononuclear cells and was able to culture them for a long period of time in the same signaling systems that I just talked about, with the addition of needing to sort about weekly to keep enriching for the immature erythroblasts that are larger. And she was able to find that this expansion of these self-renewing erythroblasts, or as we call them, induced self-renewing erythroblasts, ISREs, expanded extensively, maintaining this immature appearance that is hallmark of a erythroblast over time during expansion. And importantly, it also maintained normal karyotype during expansion and dependence on the cytokines and BMI1 overexpression. So we wanted to apply this system to the ISREs generated progenitors that I talked about. And we found that we also saw an expansion of cells that had the morphology of immature erythroblasts. They also maintain high levels of expression of BMI1 with this vector. And importantly, they express fetal pattern of beta globins. So then we needed to take those cells and mature them to functional red blood cells. I have two different medias for that we've used. One is one that Jamie had been using in the adult system, which is a relatively simple system, but recently we've also turned to a maturation medium that's a little more complex, and it comes out of our CHOP collaborators, Stella Chow's lab, with the addition of RU486, because what's important for this maturation is that we remove dexamethasone signaling. And so RU486 inhibits that signaling, and we keep that in for 1 day only. We also keep SCF, which helps earlier stages of erythroblasts in early, and then remove it earlier. So when Jamie tried her system with the adult-derived SREs, what she found was that they did go through the morphological changes that we associate with erythroid maturation, including 50% enucleation in her cultures. There are two markers that are used in the field now to sort of distinguish between immature and mature erythroblasts. Mature erythroblasts are designated as those that are CD49D low and band 3 high, which is what these cells became. Most importantly, when we sent these cells to Connie Westhoff and Julia Ashelman in New York Blood Center, along with donors' primary RBCs, we get the same results when we type with either primary RBCs or with ISREs derived from that same donor. So these worked as reagent red cells. So we've applied that to our own IPS-derived SREs, and we have also seen signs of morphological maturation, a maintenance of a fetal expression pattern. But we need to also be able to take these cells and freeze them and thaw them if we want to be able to have them be useful and to be able to be sent around. So we've done that, and we've actually had good success with being able to thaw our cells, see that they expand, move them into the maturation media, and have them undergo maturation. It's even more impressive, I think, if we use our new maturation media derived from the CHOP system, in that we can again expand for a few days and then take those cells and undergo a good level of maturation. Notice they're still nucleated. We only get about a 5% enucleation rate though. We do maintain a fetal pattern of a definitive pattern of gene expression, although with this line, we see a large amount of HBB. So we're repeating this to see if this still occurs, but this is still the definitive pattern, even though it looks a little more adult. Importantly, we see that CD49D is low and that band 3 is high on these cells, especially if you compare them to normal RBCs. And furthermore, if we look for RH, which is a really important antigen for blood typing on the surface of these cells, we see that they have very high levels of it. So we're really excited to then now very soon send these to our collaborators to see if they can be used as functional reagent red cells. And we have high hopes for that. Even though that they don't have a high level of enucleation, in Stella Chow's lab, they've been able to even use primitive nucleated erythrocytes, erythroblasts that they've derived from IPS cells and been able to use them in these reagent cards. as long as the cells get small enough. And we believe we've achieved that with the maturation protocols we're using now. So we hope to have good success with this. So in conclusion, the iPS cell system differentiates and generates a mix of primitive and definitive erythroprogenitors. And if you overexpress BMI1 in either peripheral blood mononuclear cells or these iPS cell derived definitive erythroprogenitors, you can induce this self-renewal of this erythroblasts or these ISREs. These cells from iPS cells can be frozen, thawed, and expanded, and then will shortly undergo maturation, which includes expression of cell surface proteins. And so we hope that this will be a source for reagent red cells in the future. Now, I also want to thank past members of the lab. Samantha England is the one who discovered this extensive renewal, self-renewal erythroblast. Kim worked at the BMI regulation in the mouse. Jamie Olson, as I've shown a lot of her data, and she's got a lot more on looking at the adult system. And then Ann Kaninsky, who's been shepherding all of our IPS work. And I also really want to thank PCTC, not only for the funding, but also the opportunity to work with these wonderful collaborators. The synergy of this group has really made doing this research fun. So thank you.